Oh, today. All right, before we begin, are there any questions? Can somebody please confirm you can hear me?
We can hear you.
Thank you. Let me share my screen here. Don't think I'm sharing it. All right, today is November 18th, and we are supposed to be on chapter 14. I don't think we're there. I think we need to finish chapter 8. Let me remind you, there's going to be a quiz 5, tomorrow. Quiz 5 will cover chapter 8 and lab 9. And we haven't finished chapter 8. Boy, that's a problem, but hopefully we'll finish it today. Um, Quiz 5 is worth only about 2 points, it says on the grade book. But if you look in the quiz, you can read that I say that this quiz, I've got it graded down like this only because I don't want your grade to be inflated by points in the grade book, meaning you get to throw out one quiz. So I've got quiz 5 currently worth only about 2 points. But when you take the quiz, I will then throw out your lowest quiz and move the amount of the points of quiz 5 up to the total, which is 75 points. Any question about any of that? So quiz 5 is going to count the same amount as all the other quizzes. I just don't want to inflate your grade, and then you'll say, geez, I've got an A in the class and now I don't have to worry and all that. And then I will remove the low quiz and make quiz 5 worth what its real amount is, and then you'd see where your real grade is. So that's why I have quiz 5 currently worth only about 2 points. Any question of that?
I have a question.
Uh-huh.
Is that not going to be done until like the end of the term, or is that something you're going to just soon?
I will try to do that before the end of the term. I'm obviously behind on the grading, but um, I will change the score of quiz 5 as soon as I can. And what I'll do is I'll make the quiz 75 points and then going through and look to see which is your lowest quiz and put that score in the drop quiz column. The drop quiz column is a negative points because you're going to be counting, let's say quiz 4 is your lowest quiz. You're going to get, let's say, 40 points out of that. And that 40 points would make your quiz score too high, and it will also make you have a higher point than it really is because I'm going to take off 40 points. And how I do that is I do it in the drop quiz container, uh, drop quiz column, and that will be a negative points because you're getting those 40 points in the quiz 4 column. Is that clear? The reason why I do it that way is I don't frequently make a mistake, but especially when I was first teaching, I would simply put in a number like 0 for the student's lowest quiz, and then there would be no way to check that I got it Correct. And this way, if I make a mistake, you guys will see it, and then you can point it out, and then I can make a correction. Is that clear? So you will have, let's say quiz 4 is your lowest quiz, you'll have it added in the quiz 4 container, but then I'll remove it in the drop quiz column. And quiz 5 will be moved up to 75 points, and I will do that right before I put in the drop quiz column, meaning I try to do that on the same day.
Okay, thank you.
All right. Boy, I got a message saying stop share. Hopefully it didn't stop. Let's go on to chapter 8. Let's see where we need to be on chapter 8. I told you my computer was slow. That's it. I'll have to restart it because for some reason it's being very slow. So it looks like we're on slide 60. We need to get to slide 66 to finish um, this chapter. All right, I spoke about sexual conjugation last time, where one cell has a plasmid in it, and that plasmid codes for the sex pilus to be made by this cell. And the sex pilus will go out and skewer another cell, act as a winch to bring the two cells together. And then the plasmid, which is called the F plasmid, by the way, replicates and then moves over through the sex pilus into the other cell. And then that cell now has the F plasmid in it, which codes for the sex pilus. So this cell will then make a sex pilus, and that will change the phenotype of this cell from F minus to F plus. Any question about sexual conjugation? All right, the third way that cells may gain genetic information is by transduction, where the DNA leaves one cell and then is gained by the recipient cell. In transduction, we have a virus moving the DNA from one cell to another cell. So we're going to talk about viruses just for a little bit. I realize we haven't talked about viruses yet, but a bacterial virus can infect a cell and then put its genetic complement into the cell. Let me blow this slide up. Put its genetic complement into the cell, and then the genetic complement of the virus wants to run the rules or gain control of this cell. And an easy way for this virus to do that is to degrade those chromosomes. So one of the first genes encoded on this virus, or meaning one of the first genes made on this virus, is a enzyme which then degrades the bacterial chromosome. And then only the viral genome is running the shots in this cell. The virus will then replicate its viral genome, making multiple copies of it, meaning the reddish purple there. And the viral genes will be transcribed, mainly making, but it's making the viral proteins, including the proteins which make the protein coat of the virus, which stays outside the cell, by the way. And then the virus will replicate inside this virus cell. And the last thing it'll do is it will cause this cell to lyse, and that will release the newly replicated viruses. However, viruses sometimes make mistakes, and one of the mistakes they can make is instead of adding the reddish purple viral genome to the virus, it adds the dark purple, which is a cut up chromosome piece of the host chromosome. And the virus makes many offspring, so it's not terribly worried that every once in a while it makes a mistake. But as you can see, this one and that one, the virus doesn't have viral DNA. It has DNA from this host chromosome and cut into pieces by the virus itself. But and then when this virus replicates, it will fuse with a recipient cell, meaning it's the cell that the virus normally infects, and it will inject its viral genome into the cell. But this is not virus DNA, it's DNA of this chromosome. And then that DNA of the chromosome of this cell here can fuse with the recipient cell's chromosome, and then it can be expressed, meaning this cell has gained new DNA, and that DNA came from this virus, which got that DNA from this host cell that the virus infected. Any question about how transduction occurs in bacteria by a virus? You might be asking, does this happen in the human genome? And it very rarely happens where a virus would lyse a human cell, pick up a human piece of DNA, and then infect a germline cell, bringing this DNA from this cell here into the germline cell. So that very rarely happens in the human population. It can happen. I'm not saying it doesn't ever happen. I don't know of any examples of it, but my guess is it happens all the time in our nose cells, meaning that somebody gets a virus and then the virus makes a mistake and puts the DNA of your host chromosome into the virus, and then that virus goes and infects another person's nose cell and injects the DNA from another person into this person's nose cell. This nose cell can then pick up a new piece of DNA coming from another person. However, who cares? Because that will only be in that one nose cell, and that will never go into a germ-line cell. So my guess is this happens all the time in human nose and maybe throat cells as well, maybe mouth as well too. But my point is it doesn't happen to a germline cell, so it doesn't affect the human population. Whereas in a virus, it can happen, and obviously it affects the germline population because the germline is one bacteria. Any question about any of this? Anyways, that's how genetic information can move from one cell, a donor cell, to a recipient cell using a virus. And that's transduction. Any question about uh, the uh, the way that genes can move between different um, cells, meaning DNA can move from a donor cell to a recipient cell, and that's horizontal gene transfer, which can happen by transformation, conjugation, or transduction. Now, to state the importance of transformation, conjugation, and transduction, let me get a new document here. Oh, I forgot to mention there will be no lab today. You should be working on your unknown project today. There is no lab today. I will be logging in for 15 minutes just to answer questions, but there is no lab today. Let me open up a new blank document. Actually, I can shut that down. Maybe I'll speed up my, come on. Probably not. Um, Oops. That's not what I wanted. What do I want? There we go. Make this a big print so you can see it. So spontaneous mutation happens about one cell per gene in about one, I'm going to go EE, 10 to the 6. Actually, I can go X with this. Never mind. So spontaneous mutation happens, or I'm not seeing my, so spontaneous mutation happens in about one cell and 1 times 10 to the 6 cells. And transformation in bacteria happens about one cell, and oh, I can make it right there. 1 times 10 to the 5th cells, meaning it happens 5 times more frequently. The same with conjugation and then transduction. They happen about 10 times more frequently than mutation. So that tells you how important transformation, conjugation, and transduction are to bacteria cells. That when a cell gets a new phenotype, one that it did not have, it is more likely to get it from transformation, conjugation, and transduction than from spontaneous mutation. And in fact, it's 10 times more likely to get it from one of these three than through spontaneous mutation. Any question about any of that? Let's just go ahead and close that. All right, let's briefly talk about plasmids. They are a small circular extrachromosomal piece of DNA containing genes. Extrachromosomal means that it's not part of the chromosome. The difference from between a plasmid and a chromosome is a plasmid only contains a few genes, maybe a handful, most 2 handfuls of genes, and a chromosome, as you know, contains thousands, tens of thousands, even maybe 100,000 genes, depending on the species and if there's only one chromosome in the species. So a plasmid is much smaller, but only contains a few genes. And then the plasmid can replicate independently of the cell's chromosome. And in fact, you've actually already seen that right here. The F plasmid replicated and then moved into the other cell independent of the chromosome replicating. Any question about any of that? Plasmids are important because if a cell has a plasmid, it's more likely to share the plasmid by, well, transformation or conjugation or transduction, then the cell is likely to exchange its chromosomal DNA. Any question about that? So plasmids are more likely to move from one cell to another than the chromosomal DNA is. There are different categories of plasmids. That's all you need to know. You don't need to know the names of the different categories. Just know there are different types of plasmids. All right, any questions about plasmids? If not, let's talk about a transposon. A transposon is a segment of DNA that can move from one region of DNA to another. Transposons are sometimes called jumping genes because the gene can move about on a chromosome. And in human cells or a eukaryote cell, the gene can actually jump from one chromosome to another. Any question about that? So transposon always has a transposase gene and then inverted repeats on the ends of that transposase gene. The transposase gene codes for an enzyme which actually cuts out the gene. So let's say this is attached to a chromosome here and here. The transposase actually cuts this piece out and then reanneals the DNA ends together. So the transposase gene moves out. And then the enzyme goes and cuts another piece of DNA and then inserts this gene that was cut out of that old location to the new location. The inverted repeats are sequences that help the DNA combine in the new location. So these are insertion sequences. They help for cutting and resealing the DNA, which happens by the transposase enzyme. Any question about any of that? A simple transposon, all it contains is the inverted repeat and the transposase gene. However, you can have a complex transposon which can carry around another piece of DNA, like TN5 carries around the kanamycin resistance gene. It does have, well, actually, it doesn't have any other DNA to it, but it has a transposon on both ends, one here and one there, and that's normally seen in a complex transposon carrying a piece of DNA, that it actually has a transposase here and a transposase there. And the enzyme actually cuts it here and here. And then this inverted repeat works there. And this inverted repeat works there for both removing it from the location of the DNA on this part and then helping it join a new segment of DNA that the transposase enzyme cuts the DNA. Any question about transposons? Transposons are important because they allow a gene or a piece of DNA to move from one place in the host genome to another place. It was first discovered in corn. People were trying to figure out why the corn kernels were so varied, and it turned out the color was on a transposon, and so the color gene was actually moving around, and where it was on the chromosome affected the expression of the gene and the color of the gene in the corn cell. And so different corn, where the kernels were different colors and all that. Transposons now have been found in every species, by the way, so they're very common. And they are important because, like I said, they allow the DNA to move from one place on the chromosome to another place, including a transposon can allow a gene on the chromosome to move to a gene on the plasmid, such as TN5 moving from the chromosome to a plasmid. And now this plasmid is much more likely to be shared with another cell, in which case that other cell will get kanamycin resistance gene. Any question about the importance of transposons? and how they work with a plasmid to, I don't know, spread around genes in a population. Any question about any of that? I know transposons are not something you probably ran into before, but a piece of DNA can actually cut itself out of its current location and then move to a new location. And that happens in a transposon. Any questions about any of that? All right, if not, let's talk about the last slide, genes and evolution. Genetic variation, if it's new genetic variation, it can only arise by mutation. If it doesn't exist in the species, it cannot arise until mutation creates it. An example of that was in the human population. Everybody had brown eyes, and 10,000 years ago, we had a mutant gene arise, and it was the blue eye gene in Northern Europe, arising by mutation. That gene then could spread. Obviously, it's spread by humans by sexual reproduction, but by also crossing over so that the blue eye gene can move from, let's say, the mother's chromosome X to the, well, not the father's chromosome Y, I guess, let's say the mother's chromosome 1 to the father's chromosome 1. So crossing over is important because it can then shuffle the genes up. You can get new genetic combinations where initially it was thought that the blue eye gene was linked on the same chromosome with the blonde hair gene. But by crossing over, the blue eye gene can now be linked to either the red hair gene or the brown hair gene. And it's true that the brown hair gene is not really an allele. It's a couple of alleles, but let's not get into that. But the blonde hair and the red hair, it's really a recessive gene that gives you that hair color. Any question about any of that? And the blue eye gene, which was initially linked to the blonde hair gene with crossing over, it then became linked to other genes like the red hair gene, and we'll just leave it like that. Once genetic variation arises by mutation, it can then spread in a population, at least of bacteria, by transformation, conjugation, and transduction. And these are important because a cell is much more likely to get it by transformation, conjugation, or transduction than the cell is going to get it by mutation, 10 times more likely. Transposition does the same thing as crossing over. It just shuffles the genes up, making new genetic variations possible between different genes, different alleles at different locations, I should say. All of this drives evolution, which happens via natural selection. Any questions about genes and evolution? All right, that's it for chapter 8. Any questions about chapter 8 before I leave? It's chapter 13. All right, chapter 13, viruses, viroids, and prions. The major goals and a rough outline is shown in this slide. Know the terms. What's a capsid? What's an envelope? What is a spike? What is a bacteriophage? What is the lytic life cycle? The lysogenic life cycle? What is a prophage? What is a provirus? What is the latent life cycle? What is a persistent viral infection? What is a prion? And what is a viroid? Any questions about these major goals? All right. 2, understand the characteristics of viruses, their structure, and the classification of viruses. 3, understand about viral multiplication. And then lastly, 4, understand about prions and viroids. The only goal a virus has is to replicate. How does a virus replicate? Well, it infects a host cell, invades the host cell, and then hijacks the host cell, forcing the host cell to replicate the virus. Let's watch a little video on that. We don't have a button to blow that up, but it's not terribly important. Here, maybe there it is.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
Any question about that video? All right, some general characteristics of viruses. Viruses are inert when they are not within their host cell, meaning they display little or no metabolic activity unless they are currently in their host cell. Viruses require a living host to multiply. So you can say viruses are obligate intracellular parasites. They must have a host cell in order to reproduce. They multiply within living cells using the host cell's enzymes, nucleic acids, amino acids, and etc. Viruses have no ATP generating mechanism. They simply get ATP from their host cell. Viruses contain DNA or RNA, but never both. It's a little unusual, and that is their genome can be DNA or RNA. It's unlike a cell where the genome is always DNA. Viruses do have a protein coat surrounding the nucleic acid, meaning the DNA or the RNA is the most internal molecule of a virus, shown as that bluish purple molecule here. And this is a virus with this edge cut out so you can look inside of it. And around this DNA is a protein coat. All viruses have protein coat and a genome, either DNA or RNA. Some viruses are enclosed in an outer lipid envelope, and that will be on the outside of the protein coat. So you can have a lipid envelope right there. Not all viruses have the lipid envelope. Some viruses only have the genome and then the protein coat. The host range is the range of host cells a virus can infect. This range is determined by specific host attachment sites, example like a cell wall or flagella, that the virus attaches to. The cell has to have cellular factors necessary for viral multiplication, meaning the host cell must have, let me blow this slide up.
I have a question. I thought we were doing chapter 14 this week.
Um, it might be, I have that in the syllabus. We will do chapter 14 after chapter 13.
Okay.
Okay. The host range of virus depends on certain factors called host cell receptors, and the virus will bind to that host cell receptor. There are specific molecules in the virus called ligands or adhesions, depending on, I guess, who the author is, that bind to the host cell receptor. So adhesions or ligands are molecules on the virus that bind to molecules on the host cell that we call receptors. And this largely determines the range, the host range of the virus, meaning if the virus cannot bind to those host cell, it will not infect that host cell. And there is one other thing for determining the host range, and that is the host cell has to have the factors needed for this virus to multiply. So there might be cellular factors necessary for viral multiplication, and both of these determine the host range of the virus, meaning the host cell that this virus can replicate in. Most viruses only infect the cells of about one species, and usually only a few cells of that one species. For example, HIV infects only two species, humans and one of the chimpanzees. I forget the name of it, but there are two chimpanzee species. It only infects one of them, and it only infects certain cells. Like it can infect, obviously, T cells, and it can infect macrophages, and it can infect brain cells, and a few other cells. But my point is, it only infects a few cells of even humans and the chimp. It does not infect like bone cells and nose cells. I don't think it infects skin cells. And most viruses are very specific, infecting only a few host cells of one or maybe two species. That's not true for all viruses. As you know, there are some flu viruses that hit humans, hit birds, hit horses, hit, I think, cows, and certainly pigs. And there are even a few viruses that hit cats and dogs. So there are certain flu viruses which have a very broad spectrum host range. What's another virus that'd be a good example? There's the Rocky Mountain fever virus, which hit human cells, and it can also hit tick cells, which a tick, if you don't know, is a cold-blooded insect, and it's very different than a human. At least as far as I know, all of the flu viruses, no, that's not true. The flu viruses hit mammals and birds, so it's only infecting, as far as I know, warm-blooded species, but birds are a different, what do you call that, grouping than mammals. What is that, phylum? Does anyone remember? Phyla, birds and mammals. But most viruses infect really only about a few cells of one or so different species. So they have a very narrow host range. When we're talking about viruses, you should understand that they are small. Their size ranges from about 20 nanometers to 440 nanometers. That's 0.02 micrometers to 0.44 micrometers. So the very largest virus in their diameter reaches the size and is actually slightly larger than the smallest prokaryote. It's thought that the smallest prokaryote that's viable is about 0.4 micrometers in diameter, but there are prokaryote cells which are smaller, and they are down to 0.2 micrometers, but it's thought that those are probably non-viable, the ones that are really small. So looking at the size difference, let me blow this up. Here we're seeing a portion of a red blood cell. And there's a E. coli bacterium, slightly larger than your average bacteria. The width is about the size of the average bacteria, but it's slightly longer than the average bacteria. And the very largest virus, is megavirus, which is about 0.44 micrometers in diameter. We do have a larger virus in its length, Ebola virus. It actually reaches very close to 1 micrometer in length, but it's smaller in its diameter than the megavirus because it's skinny, but it's longer than the megavirus. And these are the largest viruses. The smallest viruses get down to 24 nanometers, but most of you probably have never heard of bacteriophage MS2, one of the smallest viruses known. The smallest virus you know is probably, oh gosh, what's the name of that? Poliovirus, which is in here someplace. There it is right there, which is 30 nanometers, just a little smaller than, a little larger than the smallest virus known. So poliovirus is one of the smallest viruses we know. Down to 30 nanometers, and you can get a little smaller. You can go down to about 20 nanometers in diameter. All right, when we're talking about virus structure, you should realize that a virion is a single mature complete virus, and it's an infectious virus particle. So usually when we're talking about a virus, we will say the virus has infected a cell, and then it makes new virions, new viruses. Viruses can have nucleic acid as their genome, but that genome can be DNA or RNA. It is never both, DNA and RNA. It is either DNA or RNA. The DNA can be double-stranded, and that's normal in cells. Most of the time, our DNA is double-stranded. But sometimes the viral DNA, depending on the species, can be single-stranded DNA. And that's normal in cells too. Our cell has single-stranded DNA whenever the DNA is being replicated or the gene is being transcribed. It'll be single-stranded DNA. The viral genome can also be double-stranded RNA, and that's unheard of in our cells. Our cells do not have double-stranded RNA unless they're infected with a virus that has a double-stranded RNA genome. Our cells do not have double-stranded RNA in them. We do have single-stranded RNA in our cells, and the virus genome could be single-stranded RNA. If the virus is single-stranded RNA, it's important to say whether the virus genome is positive single-strand RNA or negative single-stranded RNA. The positive single-stranded RNA genome of a virus can be read by a ribosome, meaning the positive single-strand RNA contains the AUG codon, and the ribosome can read it. The negative strand RNA is the antisense to the positive strand RNA, and the ribosome cannot read it. It lacks the AUG starting codon. may lack other factors too, like maybe the binding site of the um, ribosome to the messenger RNA, but it definitely lacks the AUG starting codon as well. Any questions about the nucleic acid of viruses? So they can be double-strand DNA, single-strand DNA, double-strand RNA, or positive single-strand RNA, or negative single-strand RNA. The viral genome can be linear or circular. And if you remember, eukaryotes have a genome which is linear, and prokaryotes have a genome which is circular. So a virus can be either linear or circular. And then some viruses can have their genome in several segments, like influenza virus has its genome in several segments. This is actually normal in a eukaryote cell. Our genome is in several segments, and we call each segment a chromosome. You do not call a segment in a virus a chromosome because a chromosome contains protein and DNA, and the viral genome does not contain protein. So it's not called a chromosome. Any question about any of that? Let me make sure I got everything I was talking about. All right. So I've already mentioned that all viruses have a genome, which is either DNA or RNA, and they have a protein coat around that genome. The protein coat is also called a capsid. Let me blow this slide up. And the capsid is composed of capsomeres. This virus here has a protein coat here, and it is made up of one capsomere right there. Let me see if I can blow that up even further. So there's one protein capsomere right there. And then that capsomere is just repeated numerous times to make the entire protein coat of this virus. Blow that down now. Different viruses can have more than one capsomere. This virus is only showing you one capsomere. Let me show you another virus which has multiple capsomeres. Here's one. This one has one capsomere in the head. It has at least two capsomeres on the sheath, and then a third capsomere on the tail base plate. I don't know about the pin, maybe a fourth for the pin, and then a fifth capsomere for the tail fiber. So what is that? At least 5 for the sheath and then 6 for the head. So this virus has at least 6 capsomeres in it. Possibly more, but at least 6. We'll talk about the virus T, even viruses, especially T4, in just a little bit. If there is no envelope around a virus, we call this virus a naked virus. So this virus here, containing only the viral genome and the protein coat, is called a naked virus. Some viruses have an envelope around the protein coat. Let me see if I can blow that up. Such as this one here. And this we call an enveloped virus, one that has an envelope. This is a lipid layer. Let me see if I can blow that up. This is a lipid layer. The bluish region there is mainly phospholipids, but it can have proteins and carbohydrates in the envelope as well. The envelope frequently has spikes on it, which is this reddish purple spike right there. And these spikes are usually glycoproteins. The spikes are also usually antigens. An antigen is something on the outside of a virus that the host organism sees and then mounts an immune response against. So this virus will have the spike being the molecules that the host animal will see, recognize, and then mount an immune response against. And so the antibodies or whatnot that are made to attack this virus, they will frequently attack the spikes because that's the antigen. Any question about what an antigen is? Antigens are always the outermost molecule of a thing that the host is mounting an immune response to. And these spikes also serve as the attachment points for binding of this virus to its host cell. And that's because the attachment points of the virus are always the external-most points of the virus. And in that case, it would be the spikes of this virus. What is the attachment point for this virus? Another way of wording it is, what is the most external molecule on this virus?
The capsimeres.
Yeah, the capsimer or the capsid is the both the antigens and then the attachment points for this virus. And that's because the antigen and the attachment points are always the outermost molecules on the virus. Any question about any of that? And thank you for answering that, by the way. Let's talk a little bit about the general morphology of viruses. Morphology means shape. Viruses are classified on their basis of their capsid structure. We have three virus shapes based upon their capsid. We have a helical virus. Let me blow this up. Shown here, which has a helical capsid. And this, actually, the entire virus is in a helix. I don't see the helical virus in this Ebola, but it claims that this is a helical virus. And it might be that the capsid is being hidden, the helical capsid is being hidden by the envelope of Ebola. I don't know. And then that's one helical virus. The second general virus shape is the polyhedral viruses. This is like a many-sided dice, whatever that would be, many-sided dice. That edge is cut off, that should be filled in. And there we see a polyhedral actual electron microscopic image of a virus. These right here are actually spikes. But there's the envelope right there, unless that's the envelope right there, but I think that's the envelope there. And then the third general morphology, meaning the third shape, is what is called a complex virus. They can be almost any shape. Here we have T4, which actually looks like the lunar lander of the Apollo missions. And there's an actual electron microscopic image of a T4. Here we have another complex virus, and a third complex virus, and even a fourth complex virus. And as I stated, the complex virus can take on almost any shape. T4 is a naked virus, but orthomyxovirus is a complex virus. Oops, back. And influenza is also a complex virus. All right, let's talk a little bit about the complex virus T4. T4 is the virus that has been studied the most. So virologists, the people who study viruses, know more about the virus T4 than any other virus. They just decided a long time ago to intensively study T4 more than any other virus. And I think the reason why they did was simply because T4 was a virus easy to grow because it infects E. coli. And all microbiology labs have E. coli in them, so T4 was one that was chosen to be studied the most. T4 belongs to the T-even family. They all look similar, like the lunar lander here. T4 is called a bacteriophage. Bacteriophage means it is a phage or virus that infects bacteria cells. And we call a bacteriovirus a bacteriophage. So that's the correct term for a bacterial virus. I tend to use the word bacterial virus and bacteriophage synonymously, which they are synonymous. Any question about any of that? T4 does belong to a virus family. It's called the T-even virus family because T2, T4, and T6 all have the same shape. Let's talk a little bit about viral infection and the host immune response. Infection with a virus to an animal will stimulate the host animal to produce antibodies. The antibodies recognize and bind to the viral surface proteins, meaning the antibodies, as well as the host itself, will recognize the viral surface proteins that we call antigens. And that's what an antigen is, just a portion of something that the body is responding to. An antigen is almost always the external most molecules of the thing infecting the human or animal. And obviously, not all viruses infect animals, but for the moment, we're only talking about that. I don't think plants make antibodies, so it's not terribly important either. Only animals make antibodies. If the host can respond to certain parts of the virus and then mount an immune response against those parts, you can imagine that the virus might want to change those virus parts if the virus parts come under attack. And some virus proteins can change and mutate frequently. It depends on the virus strain. There are different viral strains. And if the virus can mutate or change frequently, you can have different viral strains that can therefore infect the same individual more than once. Can anybody give me an example of a virus that changes or mutates rapidly, or you can have it, the viral infection more than once?
The flu.
Yeah, the flu, COVID-19, another good example. Both of these viruses have their outer molecules changing fairly rapidly. And that's why they recommend you get a flu shot every year, which, by the way, I need to get my flu shot this year. Haven't done that yet. It's because the flu can change or mutate rapidly. The same thing can happen with COVID-19, and that's why the initial vaccine for COVID-19 probably no longer will prevent you from getting COVID-19, the initial vaccine. It may protect you from getting a serious COVID-19 case, but it will not protect you from being infected with COVID-19, meaning you can get COVID-19 again because the virus has changed over time and the original vaccine it's certainly not as good at protecting you from the um, the current version of COVID-19 out there. But the old version of the vaccine will protect you against the old version of COVID-19. So you will not get the old version of COVID-19. And for this reason, the medical profession has been making new versions of the COVID-19 vaccine. And the latest is always a little bit behind the virus, meaning the virus is continuously mutating, but it's recent. And so the newest version of the COVID-19 virus, which I think just recently came out, should protect you from getting infected from the most recent strains of COVID-19. So you would not get it if you had the vaccine, and you should not get it more than twice from the current version. The trouble is COVID-19 will continue to mutate and change, and there will probably be a future COVID-19 variant which can then infect you. Not all viruses mutate or change frequently. For example, the polio vaccine was invented, both of them, in the 1950s. I think one was 1954 and the other one maybe 1956, but don't quote me on that one. I don't remember the year. And we're still using the polio vaccine today because polio has not changed, and the old vaccine made in the 1950s is still good. And what is that? That's 75 years ago. That's a long time. As you know, COVID-19, a new variant comes out about every 6 months. And the flu, I don't know how frequently a new variant comes out, but they say that you should get the flu shot, a new one, about every year to protect you from getting the flu. Any questions about any of that? So some viruses can change and mutate rapidly, but not all viruses do. And frequently a lot of them, like smallpox, of course that one's extinct now, but polio. Trying to think of another vaccine, rabies. A lot of these vaccines were developed years ago, and they're still good. I don't know if the rabies vaccine was actually developed by Louis Pasteur or if they made a new one. But if it was made by Louis Pasteur, it was made more than a century ago. All right, let's talk a little bit about viral taxonomy. We do group viruses into families based upon the type of nucleic acid, the replication strategy, their morphology. You can group viruses above a family. Let me see if I can remember what that is. King Peter's son came over from, so that's a family. So there is an order to a virus. So most viruses have an order, but above an order, most viruses are not classified. We do not classify a virus into the phyla, the subphyla. Some viruses have a class, I think. Maybe it's order, and then all viruses have a family. Why do we not classify viruses higher, much higher than a higher than an order? Is because viruses, their genome is pretty small, and the genome changes rapidly to evolve with the host cell it infects. And so the viral genome is not very large, and then it also changes rapidly. And that makes it difficult for us to classify viruses much above an order. It's just their genome is not large enough we do not know how viruses evolved and where they evolved from, and that's why we do not classify them higher, much higher than an order. There is one exception, though, and that is we can classify a virus into the family, not the family, into the kingdom that they infect, meaning we have animal viruses, they infect animal cells, and we have plant viruses, they infect plant cells. We don't know the genetic relationship, but we do know the cells that the virus infects. And usually an animal cell will not ever infect a plant cell, and a plant virus will not ever infect an animal cell. Any question about any of that? All right, I already mentioned all viruses are classified into families. All viruses are also classified into a genus underneath that. And then they can be classified into viral species of the genus. And then we have a subspecies of the virus. A subspecies will have the same name as the viral species, but it'll be designated by a number. The family name of a virus will end in viridae. The genus name will end in virus. The virus species will have a common English name. For example, the poliovirus is called the poliovirus worldwide. There is no Latin designation for viruses, meaning no Latin naming system. Everywhere in the world, the poliovirus is called the poliovirus, and it's simply because the first people studying viruses were English speaking, and then they tend to give the virus their names. And then the other people who studied virus, the French and the Germans largely, but other groups as well, just use the common English names. I don't know if that makes you happy or not, but you don't need to know any Latin naming taxonomy for viruses because they're all using the common English name. Any question about any of that? Let's talk about how important the subspecies is to a virus. We have poliovirus subspecies 1. It infects the human and then causes the disease polio. We also have the subspecies poliovirus 2. This can infect a human cell, but it does not cause the disease polio. We use this virus as the live polio vaccine, meaning we give poliovirus subspecies 2 to people to prevent people from getting the disease polio. So that poliovirus subspecies 2 is used as a vaccine to prevent getting the disease polio. That tells you how important the subspecies is and how different the subspecies can be. Any question about any of that? Another subspecies that you know, besides the poliovirus 2, which is called I think that's called the Sabin vaccine, and then poliovirus 1, which causes the disease polio. You also probably know herpes simplex type 1 and herpes simplex type 2. Both of them are herpes simplex virus. They infect human cells. They can infect the same human cells, by the way. Herpes simplex type 1 can infect your genitals, and herpes simplex type 2 can infect your mouth, but usually herpes simplex type 1 infects your oral mucous membranes in the face, causing a cold sore. And then usually herpes virus type 2, subspecies 2, infects your genitals and causes the herpes virus sexually transmitted disease. Once again, telling you the importance of the subspecies. Any question about any of that? All right, let's give an example of the viral taxonomy. An example is the family Herpes viridae, the herpes virus family. The genus can be the herpes simplex virus, which the subspecies can be herpes simplex type 1, causing the cold sore. I'm not going to talk about the common name, the human herpes virus, because it's more commonly called the human herpes simplex virus. I mean the herpes simplex virus type 1. The this name has not stuck. Even medical professionals don't tend to call it the human herpes virus. They tend to call it the herpes simplex virus type 1. Any questions about that? So the family name ends in viridae. The virus, the genus name, simply ends in virus. So the herpes simplex virus. And the subspecies, herpes simplex type 1. How do you grow bacterioviruses in the lab? Remember, a bacteriovirus is called a bacteriophage. They must be grown in a host cell. And what you do is you make a lawn of bacteria on a plate. Let me blow this slide up. And that's why this plate looks whitish in this area here. That's from a lawn of bacteria growing everywhere. And then once you put the bacteria cells on, but before you incubate the plate, you put on a solution containing the viruses, and this virus can infect the bacteria cell, and then it will cause that bacteria cell to replicate the virus. That'll cause the cell to lyse, and then who will get infected? The nearby bacteria cells. And then they will get infected and cause the cell to replicate the virus, and then the cell will lyse, and where will it go? Neighboring cells once again. And so you can get this happening, and that will form eventually a plaque on this plate, where initially there was one virus, and it replicated to lyse the cells, which then went and lysed more cells around it, causing these plaques. And you can count how many plaques are in this solution. This is a large number, maybe 100, 200 different viruses in solution here, each of them causing a plaque. Let me blow this up even further. This plaque is very large, and it looks like it's three plaques that merged together, but that plaque is not, and that plaque is not, and that plaque is not. Why is that plaque larger than, let's say, that plaque there? Or that plaque even there? Why is that plaque larger and that plaque larger? It could be that when the virus solution was first put on the plate, the virus initially attached to a cell and then was replicated immediately in this bacteria cell here, which then grew into a large plaque here and here and here, meaning that the virus initially infected a cell and then replicated. But the smaller plaques, the virus went down near the cell. So here's our virus, and then here's a cell nearby, and the two didn't touch. We needed this virus to replicate again, in which case it's replicating here and here. And we needed it to replicate once more before the bacteria cell was touching the virus. Now that's touching, and so the virus can infect that cell and then cause the cell to replicate the virus, causing this cycle of the viruses to lyse the cell and then growing into a plaque. And that could be a reason why some of these plaques are smaller than other plaques, like that plaque there, that plaque there. They're smaller than these plaques here. Any question about any of that? Anyways, I used to grow up viruses when I was an undergraduate and graduate student, and that's how I grew them up. Remember, for a virus to multiply, the virus has to hijack its host cell and cause the host cell to replicate the virus. The reason is the viral genome only contains a few of the genes needed for virion synthesis. The virus relies upon many factors from the host cell to replicate. And the virus will take over the host cell's metabolic machinery using the host cell's ribosomes, the host cell's tRNA, the host cell amino acids, the host cell's ATP, and some of the host cell enzymes needed to replicate the virus. The basic mechanism for viral multiplication is similar in all viruses. And that is the virus infects the cell and then causes that cell to replicate the virus. I think I'm going to stop here. Well, I guess I can talk about this slide. Bacteria viruses have only two means for replicating. Is that true? Well, they have two main ways of replicating. They have the lytic life cycle and the lysogenic life cycle. In the lytic life cycle, the virus attaches to its host cell, uses the host cell to replicate, and then will lyse the host cell. So the host cell is killed. In the lysogenic life cycle, the host cell remains alive for as long as the virus remains in the lysogenic life cycle. And these are the two main ways that bacteriophages can replicate. Any question about any of that? If not, I'll stop here and we'll continue with this in our next lesson. Any questions? All right, I'll be in the lab for 15 minutes to answer questions, but after that, I'll log out. Bye.